In our second SoftChalk lesson of Lecture 3 in Unit 1, we're going to be looking at the gram-positive cell wall, also known as a cell envelope. And this is a little longer SoftChalk lesson because we're going to start relating these various anatomical components of bacteria, like their cell walls, to various processes we're concerned with during the course. We want to see what these structures, like the cell wall, do for the bacterium, but we also want to look at their role in initiating body defenses. Components of the bacteria are what are going to eventually initiate both innate immune defenses and adaptive immune defenses against the bacteria. We want to take a look at the role of these anatomical structures, like components of the cell wall, in their role in pathogenicity. That is, their ability to enable bacteria to colonize the body and harm the body. So these are all topics we'll be taking up later on in the semester in greater detail. Unit 3 deals with bacterial pathogenesis. Unit 5 deals with innate immunity. Unit 6 deals with adaptive immunity. But we want to start introducing some of these relationships here and some of the terminology so that when we get to these units coming up, we have a little bit of background information on them. So there's quite a few fundamental statements in this one. Starting out with, because of the nature of their cell wall, gram-positive bacteria stain purple after gram staining. The gram-positive cell wall consists of many interconnected layers of peptidoglycan, and gram-positive bacteria do not have an outer membrane like gram-negative bacteria and acid-fast bacteria do. The function of the peptidoglycan, as we learned in a previous SoftChalk lesson, prevents osmotic lysis in a hypotonic environment where most bacteria live. Also found in the gram-positive cell wall are teichoic acids and lipotechoic acids. These are interwoven through the peptidoglycan layers. There are surface proteins embedded in the cell wall, and they can have a number of functions, including functioning as adhesins, be involved in secretion systems, or function as enzymes. We're going to see that the gram-positive cell wall activates both the body's innate immune defenses and adaptive immune defenses. We'll see that the body activates innate immune defenses by recognizing molecules unique to microorganisms that human cells don't have. And these are called pathogen-associated molecular patterns, or PAMPs. And these PAMPs on the bacteria are going to bind to pattern recognition receptors on their defense cells to trigger the production of inflammatory cytokines. So during innate immunity, the PAMPs binding to the PRRs are what's going to trigger inflammation, and that's how our body delivers defense cells and defense chemicals to the infection site. So inflammation, we're going to learn, is a critical part of body defense, activated by recognizing components of bacteria. But excessive inflammation can be very harmful and even fatal. PAMPs that are associated with the gram-positive cell wall are going to include peptidoglycan monomers, teichoic acids, lipotechoic acids, and mannose-rich sugar chains. We're going to learn a little about adaptive immunity. An antigen is a molecular shape that reacts with antigen receptors on lymphocytes to initiate adaptive immune responses. The actual part or fragment of an antigen that actually reacts with antibodies and with receptors on B and T lymphocytes are called epitopes. We're going to see that innate immunity is an antigen non-specific defense that the host uses immediately or within several hours after exposure to a microbe. It's the immunity we're born with, and it's the body's initial response to eliminate microbes and prevent infection. Whereas adaptive immunity is antigen-specific defense mechanism. It takes several days to become protective, and this is designed to react with and remove specific antigens. And it's called adaptive because we develop that throughout our life based on what we're exposed to. There are two types of adaptive immunity. Humoral immunity involves the production of antibody molecules in response to an antigen, where cell-mediated immunity involves the production of cytotoxic T lymphocytes, activated macrophages, activated NK cells, etc. And these are what we use mainly to get rid of infected cells and cancer cells. And then cell wall molecules can trigger adaptive immunity, such as the production of antibody molecules against cell wall antigens. And we can produce opsonizing antibodies that will bind bacteria to phagocytes for more efficient phagocytosis, as well as antibodies that bind to cell wall adhesins to block them from attaching to host cell receptors. So a lot of the stuff under immunity, we're just getting some basic definitions down now, and we'll be doing this in several parts of this Unit 1 so that those terms become familiar with you. So when we get to Unit 5 and Unit 6, you have some of this terminology down. And we begin to visualize how we defend ourselves against microorganisms with our innate and adaptive immune responses. So anyway, those are the bullet points we want to look for as we go through here. And we have a fair number of detailed learning objectives. You should be able to state what color gram-positive bacteria stain after the gram stain procedure. Describe the composition of a gram-positive cell wall and indicate the possible beneficial functions of the peptidoglycan, teichoic acids, and surface proteins. In terms of innate immunity, define pathogen-associated molecular pattern as well as pattern recognition receptor. Briefly describe how PAMPs, pathogen-associated molecular patterns of the gram-positive cell wall, can promote inflammation. State the function of bacterial adhesins, secretion systems, and invasins. Compare and contrast innate immunity and adaptive immunity in a very general way. In terms of adaptive immunity, define antigen and epitope. In terms of adaptive immunity, define humoral immunity and cell-mediated immunity. And briefly describe how opsonizing antibodies can promote phagocytosis and how antibody made against cell wall adhesins can block colonization. And also, this is the first SoftChalk lesson where we have a highlighted bacterium. So as you go through some of the SoftChalk lessons in Unit 1 and then later in Unit 3 when we come back to bacteria in more detail, we're going to be giving you a few highlighted bacteria. We're especially going to look at those that are involved in healthcare-associated or hospital-acquired infections. So when you come across one of these highlighted bacterium in the SoftChalk lessons, you want to click on that link, and that'll give you a description of that bacterium. Its gram reaction, its shape, its arrangement, its normal habitat, how it's transmitted, and the infections it causes. And so by going through that and making yourself a little note card or whatever on that, you should be able to match up the description I give you in the test with the name of the organism. And when we get to the first of these, I'll kind of give you an example of how that works, but after that, you'll just pretty much know how to do that. So those are the bullet points and the detailed learning objectives for the gram-positive cell wall. Now, as we said, sometimes it's called a cell envelope because it envelopes the cell. In other cases, it's called a cell wall. Sometimes a cell wall refers to the peptidoglycan, but I like to just call it all collectively the gram-positive cell wall, which means the same thing as the cell envelope. So as we mentioned previously, gram-positive bacteria are those that retain the first dye in the gram stain procedure, a purple dye called crystal violet. And when we're done staining them, they appear purple under the microscope, like this gram-positive staphylococcus that we see here in Figure 1. And in Lab 6, we'll go through the gram stain and see what's involved in that. And so there's a number of important gram-positives we'll be looking at in the semester, including Streptococcus pyogenes that cause a strep throat, Streptococcus pneumoniae that is a frequent cause of pneumonia, ear infections, sinusitis, Staphylococcus aureus, a common cause of wound infections and abscesses, Enterococcus faecalis, some fecal strep that live in our intestines that often cause healthcare-associated infections, and Clostridium species, to name a few. And here's our first highlighted bacterium, Enterococcus, which we're also going to take up in lab 14. So whenever you encounter one of these, click on that link. And may have to right click on that one. Let me give that a try. Okay, so if it doesn't get it with a click, right click on it. Anyway, here's a little description of Enterococcus. So what I would give you would be a description of the organism. Like I would say it's a gram-positive coccus occurring in pairs and short chains. Well, just that part alone is going to narrow it down to just one of several in this unit. So that itself narrows it down quite a bit. And these are normal flora of the intestinal tract in humans and animals. And the source is usually the patient's own fecal flora getting where it shouldn't be, although the feces from one patient can be transferred to another patient by people coming in contact with that patient. It's responsible for about 10% of hospital-acquired infections or nosocomial infections. It's a fairly common cause of urinary tract infections and bacteremia, where bacteria get in the blood. It can cause endocarditis. It can cause wound infections, especially trauma to the intestines, where bacteria get out of the intestines into the peritoneal cavity, causing abscesses inside the peritoneum, especially with things like gunshot wounds, knife wounds, surgical wounds to the intestines. From the urinary tract can get up into the kidneys and cause prostate infections. It can cause infections of diabetic ulcers, decubitus ulcers, or bed sores, burns, surgical wounds. So there's a lot of things it causes, but I would give you a list of the types of infections it causes, which is basically a result of fecal bacteria getting where they shouldn't be in the body. So given that description, you'd match that up with the genus Enterococcus. Now let's look at the structure and composition of the gram-positive cell wall. So when we see it in an electron micrographs, the gram-positive cell wall appears as a very broad, dense wall, maybe 20 to 80 nanometers thick. A nanometer, remember, is a billionth of a meter. And it consists of numerous interconnecting layers of peptidoglycan. So it's often many layers of peptidoglycan stacked on top of one another. In fact, 60 to 90% of the gram-positive cell wall is just peptidoglycan. And it's thought that it's laid down in cables, and these get cross-linked all together and forms a very strong cell wall. So if we take a look at our electron micrograph in figure 2A here, this is the cytoplasm inside the cell. The railroad tracks are the cytoplasmic membrane. The space between the membrane and the wall is called periplasm. And this is the cell wall. So notice a gram-positive cell wall looks very thick and dense, surrounding the cytoplasmic membrane. Now, if we look at the illustration of that in figure 2B, we see that it looks thick because it's many layers of peptidoglycan all connected together. Now, also we're going to see that interwoven amongst that peptidoglycan are both teichoic acids and lipoteichoic acids. The lipoic acids tend to wind through the peptidoglycan and attach to the cytoplasmic membrane. The teichoic acids just kind of are woven through the peptidoglycan. And we're also going to see that all bacteria have unique surface proteins, unique to that organism, that carry out various functions. But anyway, that's the thick-looking cell wall consisting of numerous interconnecting layers of peptidoglycan in the gram-positive cell wall. Now, as we said, interwoven throughout that are teichoic acids and lipoteichoic acids. And they bind to the peptidoglycan. They can also bind the peptidoglycan to the cytoplasmic membrane. And then the outer surface, as we showed you up here, often has surface proteins that carry a variety of functions, and these vary with the strain and species. And the space between the peptidoglycan cell wall and the cytoplasmic membrane is called periplasm. And actually a gelatinous material in the periplasm between the cell wall and the membrane. So that's what the structure is composed of and what it looks like. Now let's take a look at what these various components do for the bacterium. So the functions of the gram-positive cell wall components. Well, we learned in a previous SoftChalk lesson what peptidoglycan does. It prevents osmotic lysis, prevents bacteria from bursting when they live in their normal hypotonic environment. The teichoic acids and lipoteichoic acids probably add to cell wall strength. The surface proteins can do a number of things depending on the bacterium. Some of the surface proteins are simply enzymes carrying out surface chemical reactions. But something we're going to talk about quite a bit during the course is that many of these surface proteins function as adhesins. And these adhesins are molecules in the cell wall that enable the bacterium to adhere intimately to our cells or to other surfaces in order to colonize cells and resist flushing. Any microbe that's going to colonize our body has to have some way of adhering to the cells so it's not flushed away. Virtually every part of our body has a flushing action. And we're going to learn eventually several ways bacteria adhere, but one of the ways most bacteria eventually adhere is through cell wall adhesins, surface proteins that fit receptors on our cells and allow the bacteria to bind to that cell, colonize that cell, and resist flushing. Now, we also know that many bacteria involved in infection can co-opt the function of our cells for the bacterium's benefit. And there's a variety of ways they can do that, but often this is done involving bacterial secretion systems that enable bacteria to directly inject bacterial molecules called effector molecules into the cytoplasm of our host cell in order to alter our cellular machinery or our cellular communication for the benefit of the bacterium and the detriment to us. And one of the most common types is called a type 3 secretion system. We'll learn more about these secretion systems in Unit 2. But the type 3 secretion system in the cell wall produces a hollow needle-like tube called an injectosome, because it allows the bacterium to directly inject molecules from its cytoplasm into our cytoplasm. So often some of these surface proteins are involved in producing secretion systems such as injectosomes. And then in the periplasm, the space between the cell wall and the cytoplasmic membrane, that usually contains enzymes for nutrient breakdown so the nutrients can then cross the cytoplasmic membrane, typically by active transport. So in figure 3 here, we see a gram-positive bacteria, and the little circles on the streptococcus here represent proteins in the cell wall called adhesins, which bind to receptors on our cells. And that allows the bacteria then to colonize that cell and resist being flushed away so they can continue to adhere to cells and spread out over cells as it replicates. And here's a little animation showing bacterial adhesins. So again, these aren't structures, they're just proteins in the cell wall. And we see the cell wall adhesin has a shape that fits a receptor on our cell, and that's what allows that bacterium to colonize that cell. And what the bacteria usually do with these proteins is that these proteins mimic the shape of something that's supposed to bind to our cells, and that enables the bacterium to bind to our cells. But once it's attached, then it can't be flushed off as easily. So that's one of the first steps in the process of disease or developing normal flora or microbiota, is the ability to adhere to cells. And again, often adhesins play a role initially in that process. In terms of the type 3 secretion system, one of the things they can inject sometimes are molecules called invasives that enables bacteria to invade our cells. Now, most bacteria don't get into our cells. They grow on the surface of our cells. But some bacteria are able to invade our cells. And if a bacterium can get inside our cell, it can resist body defense molecules like antibodies. And it gets free food because we're already bringing the food into our cell where the bacteria are, so it doesn't have to compete with our cells for the same food. And we'll see examples of that in Unit 3 when we get to bacterial pathogenesis. But this is a little quick animation showing you how bacteria can first use adhesins to adhere to the surface of cells. And then they can use a type 3 secretion system to polymerize a hollow needle that penetrates our cell, and then they can inject effector molecules from the bacteria into our cells. And there's many different types of effector molecules that do different things for the bacterium. These particular ones are called invasives because they're going to trick our cell, the host cell here, into engulfing the bacteria and bringing it in. Even though this is not a defense cell like a white blood cell, the bacterium can trick that cell into engulfing the bacteria and bringing it into the interior. So these invasives typically cause actin to polymerize, part of our cytoskeleton, so pseudopods form. The bacteria are engulfed and placed in a vacuole and now are found inside our cell rather than on our cell, where they can grow and replicate. So those are the components of the gram-positive cell wall and what they do for the bacterium. Now, of course, our body is designed so we can recognize when infectious agents have invaded the body, so we can mount immune defenses to get rid of them. So while we're talking about the gram-positive cell wall or cell envelope, let's take a look at the role the cell wall can play in initiating body defenses. Now, our body has two immune systems. We're going to have one unit on each of these later in the semester. Unit 5 is innate immunity. Unit 6 is adaptive immunity. But we need to know the definitions at this point, not at that point. So innate immunity is what we say is antigen non-specific. An antigen-specific defense is adaptive immunity. So in adaptive immunity, the body looks for foreign antigens. In innate immunity, we don't look for antigens. And what we actually look for are the pathogen-associated molecular patterns, as we'll see in a minute. Now, innate immunity, like we said, is antigen non-specific. And this is what our body uses immediately or within several hours after exposure to a microbe. It's the immunity we're born with. And this is our body's first defense to eliminate the microbe and to prevent infection. And if there aren't too many microbes and they aren't that virulent, sometimes innate immunity can get rid of the organism. We don't even need the adaptive immunity. Now, the other immune response is much more specific, called adaptive immunity, because this is antigen specific. We'll see our definition of antigen in a few minutes. Now, this takes several days to become protective rather than hours. And this is designed to remove a specific antigen. And we develop this throughout our life based on what we're exposed to. So those are two of the definitions we need to know that we'll be using all semester long. Now let's start out with innate immunity. How do we initiate that? Well, as we set up here, it's antigen non-specific. So we're not looking for antigens. Rather, what we're looking for in most cases are pathogen-associated molecular patterns, or PAMPs. So in order to protect our body from infection, one of the things the body does is detect that microorganisms have invaded the body. And we do this by looking for molecules that microbes have that human cells don't have. And during innate immunity, these unique microbial molecules that the human cells don't have are called pathogen-associated molecular patterns. So these are molecules associated with the microbe, or in this case, a pathogenic microbe. And their molecular patterns means they have a certain shape, like all molecules. Now, fragments of peptidoglycan act as a PAMP. peptidoglycan monomers. Teichoic acids and lipoteichoic acids act as PAMPs. Also, bacteria and many other microbes often produce short carbohydrate chains called glycans, but they're rich in either the sugar mannose or fructose. And our sugar chains don't usually have mannose associated with them. So the idea here is that we're looking for molecules that bacteria have that human cells don't have. Well, remember, of course, all bacteria with a cell wall in the domain bacteria have peptidoglycan. Human cells never have peptidoglycan. So that's a uniquely microbial or bacterial molecule that all bacteria in the domain bacteria have. And that's something we can look for. Gram-positive bacteria have teichoic acids and lipoteichoic acids, so those are PAMPs for gram-positives. Our body doesn't make these. And we don't typically have sugar chains that are rich in mannose, like microorganisms do. So this actually allows us to recognize groups of microorganisms. By recognizing peptidoglycan monomers, we can basically recognize any bacterium with a peptidoglycan cell wall. By recognizing teichoic acids and lipoteichoic acids, we can recognize any gram-positive bacterium. By recognize mannose-rich glycans, we can recognize many different microbes that have that unique component. Now, the way our body recognizes these pathogen-associated molecular patterns, or PAMPs, is that we are born with the ability to make pattern recognition receptors on our defense cells that fit these PAMPs, like pieces of a puzzle. So the PAMP on the microorganism, the pathogen-associated molecular pattern, such as, say, teichoic acid, binds to a pattern recognition receptor on a defense cell that fits that teichoic acid, and that in turn triggers innate immune defenses like inflammation, fever, and phagocytosis. We'll have a big section on inflammation throughout Unit 3 and later in Unit 5 as well, but we want to start introducing that concept now because we're going to be talking about that all semester long. Inflammation is the first response to infection or injury, and it's critical to our body defenses. We tend to think of inflammation as harmful because we feel the adverse effects of inflammation, the pain, the swelling, whatever. But in fact, inflammation is our delivery system for defense cells and defense chemicals. So most of our body defense elements, our defense cells, like the various white blood cells, are located in the blood, as are many defense molecules like antibody molecules and such. And the blood's the ideal place to have defense cells and chemicals because the blood, of course, goes to every part of the body, and there's always new blood coming along behind it carrying more defense cells and more defense molecules. Well, inflammation is how our body defense cells and defense chemicals leave the blood and enter the tissue around the injured or infection site. If the blood cells, if the defense cells and the defense chemicals just flowed past the injured site, that wouldn't do us any good. So we have to have some way to get those defense cells and defense chemicals out at the infected site or the injured site. And that's what inflammation does. So various body defense cells in the body, including macrophages and dendritic cells and other cells we'll learn about, have receptors, pattern recognition receptors that fit the PAMPs. And when the PAMPs, the molecules on the microbe, bind to the pattern recognition receptor, the receptors for those PAMPs on our defense cells, that triggers the defense cells to start producing regulatory chemicals called cytokines. And we'll be talking about cytokines all semester long too. Cytokines are the molecules that regulate every aspect of body defense. Virtually everything that is activated or turned off or enhanced or repressed during body defense is regulated by these regulatory proteins called cytokines. And what we're looking at here are a bunch of cytokines that specifically play a role in inflammation. They're called inflammatory cytokines. Now again, we'll talk about these later in the semester, but they're things like interleukin 1, interleukin 6, interleukin 8, TNF alpha and such. And so once the defense cells release these inflammatory cytokines, they bind to cytokine receptors on target cells and initiate inflammation. And they do this by vasodilation. They cause the blood vessels in that area to become more permeable so that defense chemicals dissolved in the liquid part of the blood or plasma leak out and defense cells squeeze out, and then they can go to the site of infection. So by making the smaller blood vessels more permeable, that's going to allow us to deliver defense cells and defense chemicals at the injured or infected site. Now, they also activate the body's complement pathways that we'll learn about later on. This is another part of innate immunity. And they also activate the blood clotting pathway or coagulation pathway, another part of our body defense. So these cytokines play a big role in delivering defense cells, defense chemicals to the area, activating other body defenses like the complement pathways and the coagulation pathways. Now, again, we're going to learn all about this in Unit 3, but we want to introduce the basic idea. The main part you need to get right now, and one of your objectives, is the idea that PAMPs on the microbe, in this case gram-positive PAMPs, because this is a gram-positive bacteria, bind to pattern recognition receptors on defense cells. That causes the production of inflammatory cytokines, which leads to vasodilation, increased permeability of the blood vessels, so that defense cells and defense chemicals can get out and fight the infection. And this is kind of summarized in one of the illustrations we'll look at later in Unit 3, Figure 5 right here. So let's say a gram-positive bacterium is lysed, or it releases molecules during replication, such as peptidoglycan monomers, teichoic acids, etc., which act as PAMPs. Well, our defense cells, like this macrophage, have pattern recognition receptors that fit those different PAMPs. And when the PAMP from, this case, the gram-positive cell wall, binds to the pattern recognition receptor on this defense cell called the macrophage, that causes the transcription of genes coding for inflammatory cytokines. And now those defense cells start secreting these inflammatory cytokines like interleukin-1 and TNF-alpha, and they trigger inflammation. They're going to cause increased capillary permeability so that defense cells can leave the blood and enter the tissue, and defense molecules dissolved in the plasma of the blood leave the blood and enter the tissue. They also activate the blood clotting pathway, which helps to eventually stop the person from bleeding following injury, but it also forms clots around the bacteria so they don't spread as well. And the coagulation pathway can also promote inflammation. And these cytokines also activate the complement pathways, which also promote inflammation and phagocytosis. So this is the way we kind of turn on our innate immunity, the inflammatory response that's going to deliver defense cells and defense chemicals where they're needed. PAMPs from the microbe binding to pattern recognition receptor on the defense cells, causing them to release inflammatory cytokines, and inflammation is our delivery system for defense cells and chemicals. Now, again, we'll learn the details about that later on in Unit 3 and again in Unit 5 when we get to innate immunity. So that's initiation of innate immunity, PAMPs binding to PRRs, leading to inflammation in this case. But also, bacterial cell wall components can initiate adaptive immunity. So various proteins and polysaccharides associated with the gram-positive cell wall also act as antigens, and antigens are what initiate adaptive immunity. So we define an antigen as a molecular shape that reacts with antibody molecules once they're made, but more importantly for initiating immunity, they bind to antigen receptors on lymphocytes. So the way we activate adaptive immunity is we use special white blood cells called B lymphocytes and T lymphocytes. And again, we're going to learn much about these later on. You don't have to know what they do at this point, but we'll eventually get into all the cells involved in body defense. The B lymphocytes are the ones involved in antibody production. The T lymphocytes are involved in cell-mediated immunity, and they regulate immune responses through cytokine production. We'll learn later on. But anyway, the way we recognize an antigen is that it will fit receptors on our lymphocyte. If it fits it, it's foreign to our body, something we'd want to attack. We don't recognize our own shapes because our lymphocytes don't have receptors that fit our own shaped molecules. But we don't actually recognize the whole protein or polysaccharide. We recognize just little bits of that called epitopes. So the actual portion of the antigen that fits the antibody or fits the receptors on the B lymphocytes or T lymphocytes are epitopes. Now in figure 6a, this represents a protein molecule. Remember, a protein is hundreds of amino acids connected by peptide bonds, and because certain amino acids interact with others, it has a three-dimensional shape. Well, different parts of this protein will have different orders of amino acids in different locations, and the order of amino acids can determine a shape. So an epitope on a protein antigen is typically about 5 to 15 amino acids sticking out of that protein that has a unique shape. And our lymphocytes will have a receptor that will fit that shape. So a protein might have many different epitopes on it. There could be a few amino acids here that have this shape, a different order of amino acids here that have that shape, another order of amino acids here that have this shape. But eventually we'll recognize each one of these shapes on that protein and mount an immune response against it. And in terms of human immunity, eventually make antibodies that fit that specific shape. So this is a highly specific body defense. In the case of a polysaccharide antigen, proteins are much more diverse in shape because remember there's 20 different amino acids that go into making proteins, and different proteins have different amino acids in different orders with different shapes. So lots of different shapes on a protein. Whereas a polysaccharide typically is made up of just one or two sugars. So the epitope for a polysaccharide might be 3 or 4 sugars branching off a polysaccharide antigen like in 6b here. So again, there could be many epitopes on that polysaccharide, but they're all the same shape. So we don't mount as strong or as varied an immune defense against polysaccharides as we do proteins. And again, we'll get into that in more detail later. But we want to start thinking now of how do we recognize components of microbes as foreign and remove those microbes. And one of the ways is by making antibodies that fit different shapes on that unique molecule, shapes that our cells wouldn't have. Now there's two branches to adaptive immunity, humoral immunity and cell-mediated immunity. Humoral immunity is the production of antibody molecules in response to an antigen, and we'll see later on that's carried out by B lymphocytes. So these antibodies are proteins our lymphocytes make that are tailor-made to fit a specific epitope of a specific antigen of a specific organism. And there's a number of ways these antibodies protect the body, and we want to start looking at them right away from the beginning, not wait till the end of the course. So one of the primary ways antibodies help to protect the body is that when we make these protein antibodies that are tailor-made to fit an epitope of an organism, some of those antibodies can stick bacteria to phagocytes for enhanced phagocytosis. It's a process called opsonization, and those are often called opsonizing antibodies. So one of the major reasons we make antibodies is to stick microbes more effectively to phagocytes. Phagocytes can't really engulf a bacterium and kill it unless it's stuck to the membrane, because it has to be stuck to the membrane for the membrane to pull in and pinch off and place it in a vesicle. So we will eventually recognize epitopes on the surface of a microbe, like these little gray circles here, as foreign, because they will fit receptors on B lymphocytes. And so we eventually tailor make an antibody that fits that shape. Now, antibodies are typically Y-shaped macromolecules composed of 4 protein chains, but the tips of the antibody, called the FAB for antigen binding fragment, those parts are tailor-made to fit a specific epitope, and only that epitope. The other part's called the FC part, which does some other things. So once these antibodies are made, the FAB part of the antibody, the antigen binding portion, fits the epitope and binds to it like we see here. Now these are opsonizing antibodies, so the other part of the antibody, called the FC part, sticks to receptors on phagocytes. And so the antibody then acts like a superglue to stick that bacterium to the membrane of the phagocyte so it can readily be engulfed and eventually killed with lysosomes. So here we see pseudopods going out, and the FC receptor on the phagocyte binds to the bottom part of the antibody, the FC portion, and that sticks the bacterium to the surface of the phagocyte, and eventually it'll be engulfed or phagocytosed, placed in a phagosome, and destroyed with lysosomes, as we'll see later in the semester when we get into phagocytosis. But that's one of the key ways antibodies protect the body, is they can stick microbes to phagocytes. And another thing they can do is prevent bacteria from adhering to our cells. Remember, we pointed out that many bacteria colonize cells with cell wall adhesins, where these surface proteins in the cell wall have a shape that fit receptors on our cell, as in this animation. And that allows the bacterium to lock onto the cell so it's not flushed away, so it can colonize and replicate. But if we can make antibodies that fit specifically epitopes of those adhesins, then if the antibody binds to the adhesin first, that prevents the bacteria from adhering to host cells, and so that can resist attachment and enable them to be flushed away. And of course, if those were opsonizing antibodies, that would also could play a role in opsonization. So as we go through the course, we're going to look at some of the different ways these antibody molecules actually protect us when we make them during adaptive immunity. But again, they're tailor-made to fit specific epitopes of antigens on organisms. Now, the other type of adaptive immunity is called cell-mediated immunity. Humoral immunity is a molecular response. The antibodies are molecules, whereas cell-mediated immunity is a cellular response. Now, as we're going to learn later on, this is primarily the production of cytotoxic T lymphocytes in response to an antigen. And cytotoxic T lymphocytes are the cells we use primarily to remove infected cells or tumor cells by causing those cells to commit suicide or undergo apoptosis or cell suicide. So we're going to learn a lot more about the cytotoxic T lymphocytes when we get to viral infections, because viruses always invade our cells, and certain bacterial infections, when bacteria that invade our cells. We're able to kill the cell that is infected with cytotoxic T lymphocytes. And when a cell mutates to become cancerous, that has mutant epitopes on its surface, and we often make cytotoxic T lymphocytes that recognize those foreign epitopes on the cancer cell and destroy the cancer cell. Other cells that are activated during cell-mediated immunity include phagocytes called macrophages, defense cells called NK cells, and of course lots of cytokines are also made to help mediate immune responses. So the cell-mediated immunity is not as important against bacteria unless they're intracellular bacteria, those that get inside of cells, because they're used more to remove infected cells and cancer cells. But we do want to introduce that term, since we will be talking about that when we get to infected cells and viruses and such. And there's a couple little self-checks you can do there. So as we see, bacterial structural components like PAMPs in the cell wall, or antigens in the cell wall, in the gram-positive cell wall in this case, can initiate innate and adaptive immunity to fight that gram-positive. But also some of these gram-positive cell wall components play a role in bacterial pathogenicity. And pathogenicity is the ability to cause disease. We see this especially when the body gets severe systemic infections, where you have many microorganisms infecting body systems. So when you have large numbers of microbes present during a serious systemic infection, very high levels of PAMPs are released, because the more bacteria, the more PAMPs are released. And this can trigger excessive cytokine production rather than an acute cytokine production involved in defense. And when you have an excessive inflammatory reaction from these cytokines, that can become harmful to the body. We're going to spend a whole lecture on that later on in Unit 3, looking at how PAMPs binding to PRRs can lead to systemic inflammatory response syndrome, or SIRS, and can lead to septic shock and circulatory collapse. But this is a plan what normally happens. There's a lot of matter of degrees that goes on here. When you have a fairly minor infection, then the PAMPs bind to the pattern recognition receptors, causing the release of inflammatory cytokines. You have a moderate cytokine production, a moderate inflammatory defense, and that sends defense chemicals and defense cells out to get rid of the infection. But if you have a large number of bacteria present, like in a severe systemic infection, then very high levels of PAMPs are released. When they bind to their PRRs on defense cells like macrophages, you get excessive cytokine production. Now, up to the end here on the left, that's the same mechanism we looked at in the previous section on PAMPs triggering inflammation. But with an excessive inflammatory response, very serious things can happen. First of all, as the white blood cells adhere to the inner wall of the blood vessels to try to go to the site of infection, they can actually dump their killing chemicals inside the blood vessel, leading to damage to the blood vessels, leakage of blood, and that can eventually result in things like hypovolemia and circulatory insufficiency. It can overactivate the coagulation pathway, and clots can start forming in the blood vessels, disseminated intravascular coagulation that we'll learn about. When this happens in the lungs, as the capillaries become more permeable in the lungs, the alveoli become filled with plasma and blood, which prevents gas exchange. That's called ARDS, or acute respiratory distress syndrome. And eventually that can lead to hypoperfusion, which can lead to circulatory insufficiency, lack of oxygen, lack of nutrients, and organ failure. So the same mechanism we use to send defense cells and chemicals out to fight infections are the same ones that can actually be fatal if you have a severe systemic infection. So again, you don't have to worry about all this detail here. That's what we're going to get into in Unit 3. But I do want you to realize at this point that we use the inflammatory response to send defense chemicals and defense cells out to remove the infection. But an excessive inflammatory response can lead to serious harm to the body, tissue damage, lack of delivery of nutrients and oxygen, and eventually circulatory failure. And there's a little quick self-check you can do there. And finally, at the end here, we have a self-quiz covering the whole gram-positive cell wall. Now, again, when I was talking about things like how we initiate innate immunity and adaptive immunity and excessive inflammatory response being harmful, that isn't in your objectives yet. So remember, do what's stated in the detailed learning objectives at this point. And one of them was mentioned how PAMPs can trigger an inflammatory response. And what inflammation normally does, it delivers defense cells and the chemicals to the area, but excessive inflammation can be harmful. That's all we need to really worry about now, but that mechanism will be the starting point when we get to bacterial pathogenesis from innate immunity later in Unit 3. So that is our gram-positive cell wall.